All right, this is chapter 5. This is a chapter on sensation and perception. So what I'd like to do is start off with some definitions. Uh, the first definition would be sensation. What is sensation? Sensation is really just the detection of sensory information by sensory receptors. So as you might remember, there was another use of the term receptor. We've been through this in chapter 3. A receptor in chapter 3 is a specialized protein that's in the membrane of a neuron, particularly in the dendrites, and these proteins are specialized for associating or binding neurotransmitters. So that's one use of the term receptor. The use that we're going to have in this chapter has to do with specialized nerve endings, specialized structures that are attached to nerves. They are essentially part of the nervous system, and these structures are tuned to stimuli in the environment. They are structures that are sensitive to particular types of stimuli in the environment. So that's what we mean. If you stimulate a sensory receptor, you're going to create a sensation. The term transduction is really a process of getting from the energy of a stimulus in the environment. For instance, sound energy is really just pressure fronts in the medium of the air. So pressure fronts from the medium of the air, for them to be interpreted by the nervous system, have to be transduced. You have to get from pressure fronts in the medium of the air into neural impulses. So the energy gets transduced from pressure fronts in the medium of the air to the electrical and chemical energy that the nervous system utilizes as its mode of information transmission. Now, if you take a look on page 150, you'll see that the different senses have names, essentially. The sense of hearing is referred to as audition. The sense of smell is olfaction. The sense of taste is gustation. The sense of touch is somatosensation. We also have the vestibular sense, which is the sense of the position of your head in space. So it provides information essentially about your balance. Stimulation of the vestibular sense will let you know whether you are right side up or upside down, for instance. We have the sense of body position, or the position of your limbs. That is called proprioception. And we have the sense of movement of your body through space, that is referred to as kinesthesia. And your sense of pain, your ability to detect painful stimuli, is referred to as nociception. And your ability to detect temperature is called thermoception. And so all of these senses have specialized receptors associated with them. And each of them is sensitive to a different energy in the environment. And all that information, all of these different environmental energies, get transduced into neural impulses. And once these are transduced into neural impulses, then the brain can do its work of making sense of these impulses, and the brain will do what it does in terms of making sense. Making sense of these impulses is referred to as perception. So once we recognize, once we categorize, once we have a sense of what we are sensing, then we're moving from sensation to perception. Perception involves interpretation of these sensory impulses or these sensations. Now, if you take a look on page 150 also, the term absolute threshold, that refers to the minimum amount of a stimulus that is necessary to cause a sensation. So it's the smallest amount of a stimulus that causes a sensation. And by definition, that minimum amount is referred to as the amount of energy that can be detected 50% of the time. So for instance, your visual receptors, the most sensitive ones, can detect a candle flame 30 miles away on a perfectly dark night. Under quiet conditions, your auditory receptors can detect the tick of a clock 20 feet away. All right, so if it can be detected 50% of the time, then it reaches what we call the absolute threshold, or the threshold for detection. Now, anything under the absolute threshold is referred to as subliminal, meaning that it's detected less than 50% of the time. So that's what it means. The term Lyman, Lyman is Latin for threshold, and so if it is subliminal, it is under the threshold. So subliminal means under the threshold for detection. Now that doesn't mean that sometimes subliminal messages cannot be detected. Of course, they can. It just means that by definition, subliminal messages are detected less than 50% of the time. Now, subliminal messages do have some small amount of influence on behavior. And in laboratory conditions that can be demonstrated. However, messages like backwards tracking in audio recordings, or one little frame in an entire movie is not really sufficient to be detected. And so really the general rule of thumb is if it cannot be detected, it is not influencing behavior. And so yes, it can be demonstrated under laboratory conditions, but we typically do not live under laboratory conditions. And subliminal stimuli, generally speaking, do not have any kind of significant influence on our behavior. Now, a just noticeable difference is really the amount of stimulus energy that has to be, let's just say we start out at a threshold. We are able to detect some stimulus. Okay, how much more stimulus energy has to be applied before you notice a difference? In other words, is the stimulus more intense or less intense? How much more intense or less intense does the stimulus energy have to be before you notice a difference? When you notice the difference, you have detected what we call a just noticeable difference, or a difference threshold. That's what it is. So it's just our ability to detect a change in stimulus energy. And when we do, that's the just noticeable difference, or a difference threshold. So it's just the amount of energy that has to be applied until you're able to notice a difference. Now, I referred to perception is what the brain does. The brain, after the sensory information has been transduced into neural impulses, The brain is then organizing and experiencing and interpreting. And so when you interpret sensory information, you are perceiving. So perception is an interpretation, essentially. And bottom-up processing is essentially building a perception from component sensory inputs. All right, so this is building it from the bottom up. Top down is when you have what we might call a mindset. So you're focused on something or you're thinking about something. For instance, we could say, I want you to watch this video, and I want you to watch how many times these players in white pass the basketball to each other. That gives you a mindset. You're focusing on the number of passes. And when you do that, when you focus your attention on anything, you're doing top-down processing. You are choosing stimuli in the environment on which you are going to focus your attention. Now, sensory adaptation is really just a process of becoming used to a new environment. In that environment, let's say you enter a quiet room, you may not notice the ticking of a clock until you sit in the room for a few minutes, and then you notice that there is ticking of the clock. And then if you focus your attention on something like reading a magazine, you may not notice the ticking of the clock anymore. It's still there, it's still ticking, but you don't notice it. So that is a form of sensory adaptation. Sensory adaptation also works as a habituation process. Habituation is becoming less sensitive to a sensory stimulus upon repeated exposure to that stimulus, or sometimes constant exposure. For instance, if you wear glasses, you look through the glasses, you don't really notice the weight of them on the bridge of your nose or over your ears. If they're comfortable glasses, you wear them, you look through them, and you essentially forget they are there. They're there, but you have adapted. So that is sensory adaptation and habituation are very similar ideas. You can based on constant exposure to a stimulus, become less sensitive to it. Sensory adaptation also has to do with, let's say, if you're in a dark room and you are dark adapted, and then the light is suddenly turned on, it is momentarily unpleasant until your eyes get used to the light. All right, that's sensory adaptation. Your visual system adapts to the new level of input. And then when you turn the lights back out again and the room is dark, you can't see for a while until your eyes adapt to the new level of sensory input. So our sensory systems are adapting to changing levels of input all the time. Based on where our attention goes and based on the amount of exposure, especially if it's constant or near constant, then you tend to habituate and become less sensitive to the stimulus over time. Now, I do want you to watch a video. It's by Daniel Simons. It is linked on this chapter, and it is based on one of his famous studies, and that's similar to the example, the attention example that I gave you before, focusing on the people in the white shirts passing a basketball. How many times did they pass a basketball? Well, if you are focused on that, this is top-down processing again. You're focused on something, you have instructions for where to focus your attention. And if you focus your attention on that, you will find that even if someone in a gorilla suit walks through the people passing the basketballs and thumps his chest and then walks on a total of 9 seconds, you'll find that you may not notice it. And that's called inattentional blindness. And that's something that Simons and his colleagues were able to find, inattentional blindness. So we have this top-down ability to focus attention on something, and often that means we don't focus attention on other things that may be occurring at the same time. Signal detection theory is an approach to studying the sensitivity of sensory systems, as well as the interpretations that people make, the perceptions that people have of a sensory stimuli. And the idea is a stimulus that's in a distracting background is presented. So the stimulus is at some detectable level, and the stimulus level itself can be adjusted up or down, so it's more intense or less intense, but the background can also be adjusted up or down, so that the signal is embedded in a distracting background. And so if the signal is just barely detectable, that's kind of where you want it to be, so that you can begin studying how people respond to it. So let's just say that we have a barely detectable stimulus embedded in a distracting background. Maybe it's a word on a screen, and there's some distracting, let's just say white noise, white visual noise, maybe kind of a snowy picture on the screen as well. And so your job is to detect a word that's on the screen, and just to say if it's there or not there. All right, so if you say it's there, when it's there, in signal detection theory, that's called a hit. If you say it's not there when it's there, it's a miss. If you say it is there when it's not there, it's called a false alarm. And if you say if it's not there when it's not there, it's called a correct rejection. So there we have it, the proportion of hits, misses, false alarms, and correct rejections. And based on how the signal intensity or the background intensity is adjusted, you can do all kinds of fancy experiments on whether people can detect the stimulus. And you can also, in signal detection theory, pay people for, let's say, the number of hits. So you can change the proportion of hits, misses, false alarms, and correct rejections by introducing a payoff. All right, so essentially this would be instructions where your attention goes, and let's just say that I'm going to pay you $5 for every hit, essentially saying the stimulus is there when it is. Well, guess what? That's also going to create an increase in false alarms because you're getting paid for hits. But I can say, I pay you $5 for every hit, but I'm going to take away $5 for every false alarm. So then you get the idea. You can change the payoff, the way things pay off, and change the way people are perceiving whether the stimulus is there or not, whether they're correctly detecting it. And so there are all kinds of pretty cool experiments that you can do with signal detection theory. This has been around for a long time. It's probably signal detection approach has been around for a good 80 years. Now, the Muller-Lyer illusion has to do with the perception of straight lines in the midst of right angles or near right angles. And this is shaped by our sensory environment. Sensory systems, like your visual system, is what we might call information expectant. Your visual system needs to be exposed to a rather complex visual environment in order to correctly form synapses in the visual system and make correct judgments based on the stimulation from the environment. And there's something called a sensitive period. Essentially, if you get past the sensitive period, it's like a window closes and you're no longer able to form new synapses in the sensory system as well. And so typically in human beings, the sensitive period is childhood. You know, infancy through early childhood is particularly critical for the proper development of the visual system and for the proper development of the auditory system. And if you are not seeing or hearing early on in development, then the synapses are going to essentially take on other functions where they would ordinarily take on visual functioning in the occipital lobe or auditory functioning in the temporal lobe. You may have some differences. So, for instance, there are people who are born blind whose visual cortex takes over some audio processing capabilities, so that the visual cortex becomes essentially like secondary auditory cortex. And this works with people who are born deaf. People who are born deaf, if the deafness is not corrected with a cochlear implant, will often have their visual cortex take on, or their auditory cortex take on more visual processing capability than it would ordinarily have. So all of this is related. All of this has a developmental sequence. Sensory systems have a developmental sequence, and they need proper input from the environment to develop normally. Now, when we talk about vision and audition, it is important to discuss wavelengths. Wavelengths, the visual stimuli are measured by wavelength. So visible light has particular wavelengths associated with it, and so we can have light detecting equipment, and we can separate out different wavelengths of light, and we can do this very accurately. And the same goes with sound waves. There are wavelength, sound is also measured in wavelengths. And so the amplitude of a waveform is the distance from peak to trough or from trough to peak. So the peak would just be the highest point of a waveform. A trough is the lowest point of a waveform. So the distance from peak to trough is amplitude. The distance from peak to peak or trough to trough is the wavelength. And then if you apply any unit of measurement to wavelengths, it would be the number of wavelengths per unit measurement is referred to as frequency. So amplitude and frequency are important aspects of the stimuli for vision and for audition. Now let's talk about light waves. Light is electromagnetic energy. The visible spectrum is part of the entire electromagnetic spectrum. And the electromagnetic spectrum is the spectrum of radiation. So these are electromagnetic radiations in the environment. So you can see on page 155, you have in figure 5.6 a depiction of the electromagnetic spectrum, starting with gamma rays on the left. So these would be essentially the short or the highest frequency waveforms, going to on the right, radio waves, the lowest frequency waveforms. But what we're sensitive to, what we can detect with our sensory receptors, is only a short piece of the electromagnetic spectrum, and we refer to that as visible light. So visible light is what we see, and so if you take a look, the spectrum is from about 370 nanometers to about 740 nanometers. And once we get beyond, let's say, 740 nanometers, then we are at a wavelength that is longer than red. And longer than red is referred to as infrared. And if we look at the left of the waveform, shorter than violet is referred to as ultraviolet. So our eyes do not detect ultraviolet or infrared. We're only limited to this visible spectrum. Now, with sound, it's kind of a similar idea. First of all, we measure sound a couple of different ways. We measure amplitude in units called decibels. So the highest amplitudes would be the highest number of decibels, and the lowest amplitudes would be the lowest number of decibels. And of course, you can see that the threshold is right at 0. So we essentially calibrate the measurement of decibels to whether a sound can be detected at all or not. So we go from essentially 0 decibels, where we're just not able to detect any sound, to the highest level of sound pressure that a person might be able to tolerate, which actually is more around 120 decibels, and only for a very short time. Now, an interesting thing about sound waves is once you get above around, let's say it's 90 decibels, and so you can see 90 decibels is about the sound of a food processor or an electric blender, or also around the sound of like a gas-powered lawnmower. If you're holding it out in front of you cutting grass, it's about that sound pressure level. Now, if you've ever, let's say, been pushing a lawnmower outside and you come into the house, into a quiet room, and you notice that your ears are ringing, that's not a good sign. That's called tinnitus or tinnitus, and tinnitus is a bad sign. Ringing in the ears usually means that you are at risk for hearing damage. So even for activities that are moderately noisy, like cutting grass, it is very much advisable that you wear hearing protection, that you put some earplugs in, or put some ear muffs on, or like a sound deadening headphone on, so that you protect your hearing. Long-term exposure to loud sounds damages your auditory receptors. And as you move up above, say, 90 decibels or so, about every increase, if you increase the decibel level by 5, it cuts in half the amount of time that you're able to be exposed. So if you're, let's say, 85 or 90 decibels, you might be able to tolerate that for 8 hours. But then if you move up another 5 decibels, you can only tolerate that for 4, and so forth. 120 decibels is the sound at which most loud rock bands will play. So if you are in a nightclub or an auditorium and you're near the speakers, those are going to be around 120 decibels. It's going to be a high sound pressure level, and that level can damage your hearing only after just a few minutes of exposure. It doesn't take long to get hearing damage from being close to such high sound pressure levels. So if you're in those environments and you're near the PA speakers, you absolutely have to have hearing protection or you will experience hearing loss. You can see the pain threshold is around 130 decibels, and harm, I mean just harm such as being instantaneously exposed to the loud sound is about 140 decibels. So you can see a jet engine is right around the pain threshold. And if you ever notice the airline workers who are servicing jet aircraft, they're most likely going to have foam or silicone earplugs in, and then sound deadening headsets on as well when they are working around those, because those jet engines are excruciatingly loud and they will cause hearing damage if you go out among them without any protection. Now, the characteristic of sound that's related to the frequency of the waveform is referred to as pitch. And pitch just means that essentially sound is high-pitched or low-pitched. High-pitched sounds are high frequency, low-pitched sounds are low frequency. Timbre has to do with the mix of frequency that occurs at the same time. So timbre, like you can hear the same note being played on different musical instruments. A pure waveform is not very interesting to hear. It's just kind of a sort of a monotone, an interesting sound. It's just it's a particular frequency. What makes musical instruments interesting is the primary waveform that comes through, such as a musical note, but also the overtones, the other sound waves, if you will, that occur at the same time. And so that's the timbre of an instrument. And for instance, you can think about the difference between hearing a musical note played by a flute and a musical note played by a trumpet, a musical note played by a stand-up bass. They can all play the same note, yet you can very easily tell those different instruments apart because the timbres are different. Now, section 5.3 that begins on page 157 is where this section on vision begins. And we need to look at the anatomy of the visual system. We'll start with the cornea. The cornea is the covering of the eye, and it's essentially the barrier between the eye and the outside world. And it has a shape to it, and the shape of the cornea has bearing on how light focuses. The pupil is a small opening through which light passes, and of course that can dilate or constrict, and the dilation and constriction are controlled by the muscles of the iris. The iris is the colored portion of the eye, but it's also muscle tissue that regulates the the opening of the pupil. And then light will pass through the lens, and the lens is what focuses the image onto the retina. And the retina, if you look at figure 5.10, the retina is the part of the eye that contains the sensory receptors. So the sensory receptors are located at the back part of the eye in a structure called the retina. The part of the retina that is most densely packed with sensory receptors, in particular cones, is called the fovea. So that is the point of central focus. So whenever you look at something directly, your lens is focusing that image onto the fovea. And so the fovea is the part of the retina that is best able to detect brightness, detail, and color. It is the part of the eye that is most densely packed with receptors, visual receptors that are referred to as cones. So you can see right under figure 5.10 where cones are first defined, they're specialized photoreceptors and they work best in bright light conditions, and they are the receptors that are best at detecting brightness, detail, and color. Rods are best at detecting images in low light conditions. So if you are in low light conditions, that's where your rods are really doing their work. The rods will help you dark adapt. And as long as there's a little bit of light in a room, you are able to see or at least find your way around. If a room is completely dark, you're not going to be able to see. Being able to see involves light stimulating your visual receptors. Now, visual receptors are referred to as photoreceptors. Photoreceptors essentially detect light. So, if you take a look at figure 5.11, you can see that light comes into the eye and it passes through the vitreous humor. So I'm going to jump back and forth here. The vitreous humor is the fluid in the center of the eyeball. Vitreous humor is produced and drained at about the same rate, and it circulates within the eye, and essentially it is involved in the structural integrity of the eye, as well as nutritional support for the structures of the eye. And in the middle of the eye, you can also see that we have blood vessels. So anytime we are detecting some visual image, the light has to go through this fluid, this vitreous humor. It passes through blood vessels. It passes through any cellular debris that may be in the vitreous humor. Cellular debris in the vitreous humor is referred to as floaters. These are just cells that get loose. If you see a noticeable increase in the number of these floaters, it may indicate a medical problem and that you might have blood vessels leaking. That's a very serious issue, and you need to get that checked out right away. If vitreous humor is not draining as fast as it is being produced, then pressure inside the eye will build up. A buildup of what we call intraocular pressure, and this means pressure inside the eye, is called glaucoma. And it's a very serious eye disease, and what glaucoma will do is it will put pressure on the visual receptors of the retina, and it will damage them so that they no longer function. And so glaucoma is one of the leading causes of blindness. It is treatable with medication, but it's important to get regular eye exams and get checked for glaucoma on a fairly regular basis. Okay, so back to figure 5.11. Light comes in, and you notice that at the very back of the retina, we have our photoreceptors, the very back part. Now, some species have an additional structure called a tapetum or a tapetum lucidum. And this is a reflective structure that serves to reflect light onto the photoreceptors. Humans do not have this, but cats do, for instance. So species that trend toward being nocturnal will often have this structure. So opossums, cats, deer, the proverbial deer in the headlights, these animals have a tapetum lucidum. And this just serves to amplify light so that the animal is able to see better at night, so that the animal functions better under low light conditions. So light passes through all of these structures in the eye, the vitreous humor, the blood vessels, the various other cells that the receptors synapse onto to stimulate the receptors at the back of the eye. So forward from these receptor structures, we have other structures called bipolar cells. So bipolar cells are structures that connect your receptors, the rods and cones, to the ganglion cells. All right, and you can see they have processes or poles. So we have synapses with the rods and cones, but also synapses with the ganglion cells. So there are stages to the processing. We go from receptors to bipolar cells to ganglion cells. Now there are other cells called horizontal cells, and horizontal cells connect bipolar cells to bipolar cells. But for our purposes here, the main ones to focus on would be your receptors, your rods and cones, the bipolar cells, and the ganglion cells. Now, numbers of rods and cones. We have somewhere around 120 million rods and 6 million cones. So we have way more rods than cones. We have multiple rods synapsing on a single bipolar cell. All right, so that it's like an array. Think of the arms on an antenna. Well, it's kind of like that. You have multiple rods synapsing on one bipolar cell. That is a structural arrangement that is geared toward amplification of a weak stimulus. Whereas the relationship, the synapsing relationship between cones and bipolar cells is 1-to-1. So it takes a stronger stimulus to stimulate a cone than it does the rods. Because you have multiple rods picking up a weak stimulus, you have to have a stronger stimulus to stimulate just a single cone. So anyway, cones are going to work under bright light, and cones are better at detecting detail and color. We've already mentioned that. Now, the ganglion cells are the only neurons in the visual system that have axons. So ganglion cell axons bunch up, and the bundle of ganglion cell axons is referred to as the optic nerve. So if we go back up to figure 510, the optic nerve is this. All right, so the optic nerve is this big bundle of ganglion cell axons. Now, also, the area at which the ganglion cell axons or the optic nerve exits the back of the eye, that area has no receptors. It's just a big bundle of axons. It's a nerve, and the nerve itself does not have receptors. And so that area is blind. It's called the blind spot. And there are tests for the blind spot. You can you can find your blind spot with a specialized test. These are contained in books, and you can also Google them, and you can find your blind spot. And essentially, the blind spot is blind. It doesn't detect anything. Now, what we're going to go over next is the pathway from the retina, from the optic nerve up into the brain, which is what this is. Now, what we have here is the structural arrangement of the eye. If you have an object out in the right visual field, let's say it's out here, it's going to hit the left half of each retina, and the left half of the retina is called the left hemiretina. All right, so the left hemiretina of each eye is going to project to the left hemisphere. Left hemiretinas project to the left hemisphere. The left hemiretinas are sensitive to stimuli in the right visual field. In the left visual field, those stimuli are going to hit the right hemiretina, and so the right hemiretinas project to the right hemisphere of the brain. So the right hemisphere of the brain is sensitive to information in the left visual field. Now, the optic chiasm is this structure here. Chiasm means crossover point. So what you have is you have essentially axons from the left hemiretina crossing over from the right to the left hemisphere, and vice versa, from the right hemiretina crossing over to the right hemisphere. This structure is called the optic nerve between the eye and the optic chiasm. So it's the optic nerve between the eye and the optic chiasm. Past the optic chiasm, this structure is called the optic tract. And so you can think now, if there was damage that occurred here, for instance, let's say the optic nerve on the right, for the right eye, was cut. Well, that just means that the brain would lose input from the right eye, but the hemispheres would still receive half of the information from the left eye. So the right hemiretina would still project to the right hemisphere. The left hemiretina would project to the left hemisphere. So the hemispheres would still get half of the information from that eye, but there would no longer be input from the right eye. But if you make a cut here, let's say the left optic tract, well, the left optic tract is going to mean that the left hemisphere is no longer receiving input. All right, the left hemisphere is no longer receiving input from the left hemiretinas of each eye. And so that means the person is going to be blind for information that is presented in the right visual field. So stuff on the right side of the body would not be detectable anymore. And so the left hemisphere wouldn't be processing any visual information if the left optic tract was cut. And likewise, if the right optic tract was cut or damaged. So far we have information from the retina, optic nerve, crossing over at the optic chiasm. So we have our right optic tract and our left optic tract. Now, this sort of bulbous structure at the end of the optic tract is referred to as the lateral geniculate nucleus of the thalamus. All right, so it's a part of the thalamus. And remember that the thalamus processes sensory information for every sense except the sense of smell. Sense of smell has a different pathway, but all of your other senses are processed, at least in part, by the thalamus. All right, there is projection to the thalamus, and then from the thalamus to the cortex. So here we have it, thalamus to cortex, thalamus to cortex. Now the visual pathway is a little more complicated in this, but you can see that there is a big part of the cortex that processes visual information. Certainly this is the largest sensory processing system in terms of the proportion of cortical area that is devoted to processing information. Your visual system has more cortical area dedicated to it than any of your other sensory systems. Now, also we have areas of the temporal cortex as well as areas of the parietal cortex that process visual information. So pretty much, well, your entire occipital cortex plus parts of the temporal and parietal cortex process visual information. So we have a huge amount of brain area processing visual information. Now, color vision. We'll get into theories of color vision. There are two major ones that we want to discuss. One theory is referred to as the trichromatic theory. And the idea here is that we have three types of cones that are maximally sensitive to three types of wavelengths. All right, so essentially they're called blue cones, green cones, and red cones. If you look at figure 515 or 513, you'll see that we have blue cones, green cones, and red cones, and you can see the areas of maximal sensitivity, but you can also see that these cone types overlap with each other in terms of their sensitivity, so that if you're stimulating in the area around, let's say, 500 nanometers, you're going to stimulate all three cone types to some degree. So all three cone types are stimulated to some degree. And the way we are able to achieve discerning any visible hue or color has to do with what we call additive color mixing. And this just means that we are adding light together. And if you add light together, you are maximally stimulating a particular wavelength. So if we're stimulating around 500 nanometers here, we can see that it's a sort of a greenish blue or blue-green color, and it's going to be stimulating all three cone types. But you can also see that that's not the best stimulus for, let's say, the blue cones. The blue cones are going to be maximally stimulated about here at about, say, 430 or so. The green cones about 540, 530 or 540, and the red cones you can see right around 600, or no, it's more like 570 actually, about 570 right here. So additive color mixing just means that we are mixing colors by mixing light, and if you do that, you are stimulating each of the cone types in some proportion. And so by doing this, by stimulating each of the cone types in some proportion, we are able to see pretty much any visible hue within the visible spectrum. Now, the opponent process theory is a little bit different. This idea is that we have colors arranged in what we call opponent pairs. So we have black and white, yellow and blue, and green and red. So how do we know that colors are arranged in opponent pairs? Well, we can do simple experiment, and this works pretty well. In figure 514, if you look at the fixation point and stare at it for 30 to 60 seconds, and just keep your eyes focused on it. And it helps if you get your eyes close to it and don't allow them to move. And then if you look at a piece of white paper, just immediately shift your eyes to a piece of white paper, you'll see the opponent colors. And so the opponent color for green is red, the opponent color for yellow is blue, the opponent color for black is white. And so you see something that's very different than essentially the opposite. And so these are, that's a good demonstration of opponent processes. Essentially, we're seeing after image or opponent after images. Sometimes they're called negative after images. So we see the opponent after images, and based on that, we have determined that there are these three opponent processes. Now there are people, so that's one bit of evidence right there. Just it's called color opponency, and it can be demonstrated with a simple figure like figure 514. Now another bit of evidence for the opponent process theory is the fact that there are people who are deficient in one or more opponent processes. People who are colorblind. Colorblind people typically are deficient in the green and red opponent process, and so they don't see reds and greens. They can see blues and yellows, and they can see black and white, but not red and green. So that's the most typical form of color blindness. So that's another bit of evidence for the opponent process theory, is that we have people who are colorblind. And both theories together, the trichromatic and the opponent process theory, really those theories combine to explain pretty well how human beings are able to see in color. Now, depth perception is one's ability to see how far away something is, to know how far away something is. And for depth perception to occur at its highest level, we need two eyes. Binocular cues for depth perception are just cues that require two eyes. And so you can play a little game, perhaps you've done it, you just look at a point of focus. One thing, you can just hold your finger out in front of you, about, let's say, 2 feet out in front of you, and you look at it with both eyes, and then you look at it alternately with your right eye and your left eye. So you keep one eye open, the other eye closed, and you'll see that your finger appears to move left and right as you open and close each eye. So what that means is there's a slightly different image on the retina of each eye, and your brain is comparing these images, and the comparison of these images gives the brain a number of cues to determine how far away that finger is that you're looking at. So binocular disparity is just the idea that there is a slightly different image projected onto the retina of your left eye versus your right eye. Monocular cues for depth perception have to do with, well, they just require one eye. Monocular cue requires one eye. And one we are all familiar with would be linear perspective. So you can see figure 515, a very straight road, and it looks like the lines, the white lines on either side of the road, are converging to a single point in the distance. The same works if you look down a straight railroad track. It looks like the tracks converge to a single point in the distance. So that's called linear perspective. and it only takes one eye to determine linear perspective. You may have heard of lazy eye. And this is just an inability to move the eye, essentially the muscles that move the eye, that help you to converge the eye on a single point. Those muscles are weak in one eye. And this is noticeable in children. And so the typical treatment is to put an eye patch over the good eye, the one that has the proper muscle balance that can move it well. And so if you put an eye patch over the good eye, it forces the muscles of the other eye to get with the program, so to speak. It forces them to control the eye better. And so you keep the patch over the good eye for a few weeks until the other eye catches up. And then the balance of the eye muscles will be pretty well equal, and the person will be able to converge each retina on a single point easily. But if that's not done during childhood, then a person's lazy eye just never will converge properly. And this can result in what's called stereo blindness. A person's just not going to have the typical binocular cues for depth perception. All right, so now we're going to get into the auditory system. So this begins on page 164. We can talk about the structures of the ear, and we'll start outward and move inward. So the cartilaginous protrusion outside your head is called a pinna. And it's cartilaginous. It is made of cartilage, and it is it's a biological identity marker. It's as distinctive as your fingerprints. No one's pinna is exactly the same shape. The pinna helps to direct pressure fronts in the medium of the air into the auditory canal. So that's its job, and you can assist it by just cupping your hand around your pinna if you need to localize or amplify a sound coming from a particular direction. And that works fairly well. It just helps to direct sound into the auditory canal. And so the auditory canal is essentially a little opening that terminates at the eardrum. And at this point, the energy is we could say it's kinetic energy of molecules in the air. This kinetic energy at the point of the eardrum gets transformed or transduced into mechanical energy. And so there is essentially a mechanical apparatus. We have these pressure fronts that cause the eardrum to move in synchrony with the pressure fronts, and the eardrum moves three little bones called the malleus, the incus, and the stapes, moves these little bones in synchrony with the pressure fronts. All right, so now we have pressure fronts in the air, and here we have this being transduced into mechanical energy. So this mechanical energy stimulates a little opening called the oval window. And the opening is in a fluid-filled structure called the cochlea. So the cochlea is filled with fluid, and it contains a membrane called the basilar membrane. And on the basilar membrane are little cells called hair cells. And so this fluid-filled structure, when you have stimulation on the oval window, what it's doing is essentially it's causing a little pressure front in the fluid inside the cochlea, which then deflects the hair cells in on the hair cells which are on the basilar membrane, and deflects them back and forth, and depending on the frequency of the sound. If we have, think about our place theory of pitch perception, the highest frequencies are detected at the base of the basilar membrane. So these hair cells here are going to be the ones that are tuned to the highest frequencies. And so we start with highest frequencies, and we just follow it all the way around to the tip, and the tip is stimulated by the lowest frequencies we're able to hear. Now, frequency-wise, human beings are able to hear between about 20 and 20,000 hertz. And hertz just means cycles per second. So essentially, the number of waveforms detectable from peak to peak of the waveform, or trough to trough of the waveform. We're talking about the number of times that would occur in the unit of measurement. In this case, it would be a second. So number of cycles per second. And you can see in a young, healthy human being, should be able to detect pitches at around 20,000, 18 to 20,000 usually. And this area here, since it's closest to the opening, is also most vulnerable to damage from high amplitude sounds. So loud high-pitched sounds or loud sounds of any kind are going to hit the base of the basilar membrane first, and those hair cells are most prone to being damaged by high amplitude sounds. There are certain drugs like aspirin, for instance, that can damage hair cells. So some drugs are what we call ototoxic. It just means they're toxic for the ears. And one way you know that you may have suffered hearing damage is when you hear ringing in the ears. And if you've taken a lot of aspirin to treat a fever, you might notice ringing in the ears. So aspirin is good for treating a fever. It is good as a pain reliever, but it is not good for your ears. It's not good for your hair cells. So there are certain drugs that can damage your hair cells. Certainly high amplitude sounds, especially high amplitude sounds of unexpected onset. So the sounds that do the most damage are really loud sounds that you don't know that they are coming. If you don't know they're coming, they will do more damage than if you do. There's something called the aural reflex, A-U-R-A-L, the aural reflex. And essentially, if you know a loud sound is coming, the little muscles that are connected to your ear bones, your malleus, incus, and stapes, will kind of tense up and deaden the amount of stimulation that goes to the oval window. Whereas if you don't hear it coming, there isn't that reflex won't kick in, and more damage can ensue. So there are a number of cases of people who've been damaged by loud sounds where they didn't know it was coming. Let's say like a firecracker goes off next to your head, and you don't know that's coming. That can cause permanent hearing damage instantaneously. All right, so we talked about the place theory. So I'll start with this. The place theory, it just says that different portions of the basilar membrane are different, are sensitive to sounds of different frequencies. And we said that the sounds at the base are the ones that are most sensitive to high frequencies, and the ones at the tip are most sensitive to low frequencies. There's also the temporal theory. And essentially, we have sensory neurons, but there is an activity level of that neuron. And here we're getting into patterns of firing of sensory neurons. And so the temporal theory is essentially um, related to the timing of the activity of sensory neurons. And one thing that is clear is that cells tend to fire in patterns. Sometimes we call it volleying. So if we have a cell at let's just say it's a frequency and it's a high frequency, one cell will fire, and as it essentially resets or comes back to resting, another cell in an array will fire. So essentially cells take turns firing. And so it's the timing of the firing of the cells that create the sensation of pitch. And this is how we're able to hear high frequencies versus low frequencies. So really both ideas, the temporal theory, which has to do with the timing of cell firing or the firing of action potentials by the hair cells, as well as the place on the basilar membrane. Both of those things occur and result in our perception of pitch. Now, sound localization is just our ability to determine from which direction a sound is coming. And there are cues that require 2 ears, so cues that require 2 ears are binaural, and cues that only require 1 ear are called monaural. And you can see the part I have highlighted here, the sound waves received by your ears, if they're coming from directly in front of you or directly above you, then they're going to stimulate both ears equally. And so those would be monaural cues. But if a sound is coming from one side or the other, then the sound is going to hit one ear before the other. And so that's what we have in figure 517. You can see that the sound is hitting this person's left ear before it hits the right ear. And this differential between when sound is detected by one ear versus the other ear is processed by the brain. And we do this automatically. If we hear, say, a loud sound coming from one side of us to the other, it's just automatically we tend to look in the direction of the sound. Now, hearing loss. We've talked about toxins. So there are certain toxins, certain drugs that can cause damage to hearing receptors, certainly toxins in the environments, certain solvents or metals can cause damage to your hearing receptors. Certain illnesses can cause damage to your auditory receptors. You know, when we get to types of hearing damage, let's go back to our ear picture here. Conductive deafness essentially is going to be damage to the ear bones. All right, this area here, the malleus, incus, and stapes. If you get damage to the conductive mechanism, it could be damage to your eardrum, the ear bones. Damage there is going to result in sound not being transmitted to the oval window. So we usually call that conduction deafness. And there are a variety of causes for this. Certainly the eardrum can get damaged by really loud sounds. Any of these structures can be damaged by bacterial infection. Bacterial infections can just eat away at the bones or the eardrum itself. Whereas if you have damage to the receptors on the basilar membrane or damage to the auditory nerve, actually it's called the vestibulocochlear nerve because there are two branches. You can see there's a branch coming off the cochlea and there's a branch coming off the vestibular system, which we'll talk about later. You can see the vestibular system and the cochlea are continuous with each other. And again, the vestibular system is going to be related to the position of your head, whether your head is right side up or upside down, and of course, which direction your head is turning. More about that later. So that's the difference between conduction deafness and nerve deafness. Essentially, damage to the transmission apparatus is going to be considered conduction deafness, and nerve deafness is going to be damage to the receptors or damage to the nerve. Now, a cochlear implant is an electronic device, and it has a microphone and a fancy speech processor. So these are going to be on microchips, and an electrode array. And what you can do is you can essentially place the electrode array into the areas of the basilar membrane. So if there is if the nerve is intact or the receptors are intact, then you can stimulate the receptors directly, or you can stimulate the nerve. If there's too much damage to the receptors or the nerve, then the cochlear implant isn't going to work. So depending on the cause of the deafness, a cochlear implant can treat it, and cochlear implants are improving all the time, and they are particularly useful for decoding speech. So the speech processors for cochlear implants are tuned for the frequencies associated with human speech. They are not good for other frequencies, and for instance, music. Music just doesn't work well through cochlear implants, but it does allow a person to be able to discern speech quite accurately. And then we get into, well, let's just say a baby is born and we know that baby's deafness could be corrected with a cochlear implant. Should we? Should that baby get the implant and therefore be able to hear and therefore be able to learn to speak appropriately? If a person can't hear, it's very hard to learn how to speak because we imitate sounds that we hear people make, speech sounds that we hear people make. We imitate the speech that we hear. And especially as we're growing and forming synaptic connections. We get past the sensory or the sensitive period for language development. Learning to speak is going to be much more difficult. So there are arguments. One argument is correct it as soon as it's discovered in infancy, and another argument is saying, well, wait a minute, you know, people who are deaf get along just fine. They certainly will learn to process sign language and become more sensitive to the visual environment, but deaf people get along just fine. And then we'll say, well, it's, you know, there's a deaf culture, and maybe it could be perceived as an insult to deaf culture if you just go and give cochlear implants to babies. So it's a little bit sensitive issue, but that's what this what do you think section on page 167 is about. Now we get into our other sensory systems. We have our chemical senses. We'll start with taste or gustation. So you've learned probably since elementary school about the taste primaries. We have sweet, sour, salty, and bitter, and we are able to stimulate areas of our tongues with these stimuli very easily. Now the receptors for taste are called taste buds, and they're located on the papillae, and that's those little fleshy protuberances on your tongue. Um, Taste buds are located on the papillae, and they are sensitive to any tasteable substance that is dissolved in saliva. So for something to be tasted, it has to be dissolved in saliva and bathed over um, the taste buds. Now, relatively recently, over the last 20 years or so, um, good evidence has been accumulated that there is another primary taste, maybe two. The best known of the new primary taste is called umami. And umami is Japanese for delicious. And umami, or the umami taste, is sensitive to proteins. If you like foods that have a savory flavor, if you like cheese, for instance, or broth like chicken broth or beef broth, especially with some cheese on it, then you are stimulating your umami receptors. And umami is maximally stimulated by a chemical salt called monosodium glutamate, or MSG. And so adding a little MSG to food, in fact, does boost the flavor of the food, especially the savoriness of it. And it is added to some dishes, especially in Chinese restaurants. But also it causes some undesired allergic reactions, and it's got a level of toxicity that it makes it problematic for a lot of people. So I would not recommend adding MSG to your food. You can certainly stimulate the umami taste with foods such as broths and cheeses pretty well. A food that stimulates umami very strongly might be nacho cheese Doritos. So you have cheese and you have MSG and maybe a little tomato paste or dried tomato paste along with that. All of those together pretty strongly stimulate umami. There's also a growing body of evidence that we have receptors for fat, so that we, and a lot of people are drawn to high-fat foods. There's certainly adaptive significance to that, especially in environments where the spacing between meals is uncertain. So if you are drawn to high-fat foods and you're able to pack on calories in the form of stored body fat, that can get you through the lean times. But certainly these behavioral tendencies are not good for us when food is freely available, especially high-fat food. And it just so happens that high-fat foods are often the cheapest. So you can buy high-fat foods more cheaply than healthier alternatives. But so in terms of taste, primary, sweet, sour, salty, and bitter, but also fat and umami. Now your sense of smell is another chemical sense. And if you take a look at figure 520, you can see the olfactory bulb. The olfactory bulb projects to an area called the entorhinal cortex, and that is in very close proximity to the limbic system, which is involved in processing of emotion and memory. But you can see that the olfactory receptors are passing through a bone. It's called the turbinate bone. And sometimes people become anosmic. They lose their sense of smell because they have an injury where the head maybe is accelerated forward very rapidly, and then the head hits a solid object and causes the olfactory receptors to be sheared off. And if that's the case, they don't grow back. But otherwise, the receptors can regenerate. So if they're, let's just say the ends of the receptors are damaged, they can grow back, and usually they can grow back in about 60 days or so. Going back up to taste receptors, those can be damaged pretty easily. Just think about drinking or eating something that's too hot. You can or biting your tongue and shearing off some of them. You can grow those back in about 12 days or so. So they can regenerate. So taste buds can regenerate. Olfactory receptors can regenerate, except if their axons have just been cut off by a shearing action. But they can regenerate as well. Now, for you to be able to smell something, the aromatic molecule has to be dissolved in mucus. And so the molecules are essentially dissolved in mucus and bathed across the receptors. And in terms of smell primaries, the jury's out on that. There are lots and lots of molecules that can stimulate very particular olfactory sensations. And they're called smell primaries, and the chemicals have been identified and are used sometimes as additives to products. Think about deodorants and perfumes. We know what the chemicals are, and certainly these can be manufactured in a laboratory or distilled from natural substances. But stimulation of olfactory receptors is also variable. There are variable numbers of receptors that people have, and some people have more taste receptors than others. So people who are just chock full of taste receptors are called super tasters. And super tasters are especially sensitive to bitter tastes. There are just some bitter tastes they can't stand, like bitter coffee or the taste of broccoli. So super tasters are really, really sensitive to bitter taste, but there are also people who are very, very sensitive. They have more olfactory receptors than others, and they have very sensitive sense of smell. And if you're such a person, you can certainly gain employment as a nose, as someone who can detect very small differences. Think about just noticeable differences. Think about levels, the number of molecules of a smellable substance that someone who has lots and lots of receptors, they're going to detect a smaller number of molecules as a particular scent more easily than others. So, but those people are called noses. Noses. A nose. Get back to this. Here we go. All right, here we go. So sorry about that. The video may have dropped out. I'm hoping it will stay intact. So a nose is someone who might be employed by a perfume company or a company that develops deodorants, things like that. All right, let's move on. A pheromone. A pheromone is a substance that is secreted into the environment that serves as a signal for members of one's own species. So a pheromone, when it's detected, changes the behavior of someone else who is a member of your own species. That's what a pheromone is. So it's a substance that is deposited into the external environment that serves as a signal for members of one's own species. They're very highly developed in the insect kingdom. And in mammals, they have varying degrees of development. Dogs certainly detect pheromones pretty easily. Think about when a female dog goes into estrus, and you know, male dogs from very far away detect that and show up. So that is you know, the power of pheromones. For human beings, it's a matter of some debate. There is some evidence that there may be pheromonal communication in human beings. One line of evidence is that a number of studies indicate that women who live together in college dormitories over time will have their menstrual cycles synchronize so that their menstrual periods start at the same time. And so there's some evidence for that, and the hypothesized reason for this is that there are pheromones, that something is resetting the timing of the rhythm of the menstrual cycle, and that it's probably chemical in nature. Touch, sense of touch, or thermoception, nociception. Just take a look at figure 521. There are lots of types of sensory receptors. They're located in the skin, and they're attuned to different touch-related stimuli. So some of these are going to detect pressure, some are going to detect vibration. So as the skin is displaced, it's going to stimulate these in varying degrees. And we have areas where these receptors are denser. Essentially, your glabrous skin. Glabrous is your non-hairy skin. So essentially, let's just say the bottoms of your feet, the palms of your hands, we have proportionally more sensory receptors in those areas. And for instance, just think about getting a mosquito bite on the bottom of your foot or the palm of your hand. Not only does it itch, but it hurts. You know, there's the stimulation is stronger than if the mosquito bit you in the middle of your back, where you have relatively few of these receptors, or relatively fewer. Now, pain perception is caused by a couple of different things. Pain is caused by tissue damage is referred to as inflammatory pain. Pain that's caused by damage to nerves is called neuropathic pain. Neuropathic pain is common in diabetes, for instance. Diabetics often suffer from neuropathic pain, but other disease conditions can cause neuropathic pain. But pain that comes from tissue damage is inflammatory pain. And then there are some people who just lack the ability to feel pain, and they have what's called congenital analgesia. Now let's talk about the vestibular system. The vestibular system is again, these structures, they're located next to the cochlea. They are filled with fluid, so you have your superior canal, your posterior canal, your horizontal canal. And these structures are filled with fluid, and the receptors are hair cells, just like for hearing. We also have a couple of other structures called the utricle and the saccule. And so the saccule and utricle are have hair cells in them, but they also have little structures called otoliths. And these structures in particular are sensitive to whether your head is right side up or upside down. So those structures are going to tell you whether your head is right side up or upside down, whereas the superior canal, posterior canal, horizontal canal are going to be sensitive to rotation of the head. So you can see they're oriented in different planes. So moving your head up and down stimulates, let's say, your superior canal. Moving your head side to side is going to stimulate the hair cells in your horizontal canal. So this just lets you know the position of your head at any point in time. And of course, knowing the position of your head is going to be critical for your sense of balance. Now, related to the vestibular system is your sense of proprioception. Proprioception is knowing where your limbs are, where your body parts are. And I mentioned in class, and I'll say it again, if you point at an object across the room, some distance away, you point at it with your arm extended. So stick out your index finger, point at a distant object with your arm extended, then close your eyes, lower your arm, and then with your eyes still closed, point at the object again and open them. How close are you to pointing at the object? If you're dead on or really close, then you are, well, you're dead on or really close. That means your proprioceptive system is working properly. And also this indicates that there are receptors in your muscles, your joints, and your tendons that are sending position information about the position of that limb to the brain. So that's proprioception. It's knowing where your limbs are. Kinesthesia is the perception of your body's movement through space. So that's your kinesthetic sense. And so this information, this proprioceptive and kinesthetic information, travels via the spinal column to your cerebellum. And a lot of that information is coordinated in this cerebellum so that we have a smooth flow of movement, especially for well-practiced movements. Lastly, we get into our Gestalt principles of perception. Now remember, the Gestalt principles of perception were worked out around 100 years ago, primarily by Max Wertheimer and others. Max Wertheimer, Wolfgang Köhler, and Kurt Kafka. But Gestalt principles like figure ground. What we see in figure 523 is figure and ground, and depending on whether it's figure or ground, we tend to alternate. So you can see the vase or the goblet and two faces, but you don't really see and two faces, it's or, all right? It's one or the other. You're looking at one and then maybe it switches and then you're looking at the other. So whatever is prominent for you at a given moment is figure and the other is ground. But what becomes figure and ground tends to alternate. And so the Gestalt psychologists studied a number of these figure-ground relationships. Now, other principles, proximity, is just the idea that things that are grouped together get perceived as going together. So if they're close to each other in space, then they are perceived as going together. Items, things that look alike are perceived as going together. So if you see these rows of dots and they're different colors, well, the beige dots they go together and the blue dots go together. So that's the principle of similarity. Now, principle of good continuation. You can see you have dots, but the dots are curves, and we usually see this as two different curves, and they just happen to be crossing each other. Closure is just this ability that we have to fill in the gaps in a stimulus. So if there are elements of a stimulus missing, we tend to mentally fill in those gaps. So here we have a circle and a rectangle. And finally, perception of objects. Well, think about people. People are perceptual stimuli. They're people. All right. So person perception is interesting because we tend to classify people very frequently on superficial characteristics. So just by the way people look. So things that are easily discernible, like skin color or weight, those are easily discernible, and then often there are assumptions that we make about people on the basis of these superficial characteristics, and often these assumptions are automatic. We don't even think about them, and those are called implicit associations. And when we get into social psychology, this is going to be important. And there's a test that you can take. It's called the implicit association test. And the implicit association test is a test that essentially shows you a picture of a person and a word, and the word will either have a good connotation or a bad connotation. And so the test is going to be a measure of whether you are more likely to associate a picture of a person with a good connotation or a bad connotation. So it's the rapidity at which you make these associations that is an indication of attitudes toward people in different groups that you might have, and maybe something that you haven't even thought about. So it is a measure of prejudice, but it's implicit prejudice. You can say, well, I'm not prejudiced against people in this group, and that would be an explicit statement. But implicit, if implicitly you're making associations with pictures of people in a particular group with words that have a bad connotation, then implicitly you might be prejudiced against that group. So that's how that works. It's called the implicit association test. And the implicit association test, or IAT, can be found at implicit.harvard.edu. Okay, that concludes our recording of Chapter 5.